6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Students will also have access to omics logic resources. But how does it help advance bioinformatics teaching? Omics Logic is a portal for practical and theoretical learning of bioinformatics. It takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give the student a clear path up the Bloom's taxonomy pyramid. And no matter what the user's initial experience with bioinformatics is, Omics Logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large datasets, and introduction to basic coding languages like R and Python, or even beginning a data analysis project. This also comes with an access to the Omics Logic courses, which includes courses on bioinformatics, precision medicine, multi-omics, including genomics, epigenomics, transcriptomics, as well as metagenomics, and data science that includes bioinformatics using R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. Users will also be given directions to access the T-BioInfo platform for bioinformatic processing and analysis of data. The platform includes demo pipelines, as well as data management and analysis cloud infrastructure to run bioinformatic pipelines, and different stages of analysis are performed in different sections on this multi-omics platform. So to view the various courses, example projects, and student projects, you need to first sign up on the Omics Logic Learn portal. But before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features that you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles that includes Facebook, LinkedIn, Instagram, OrcID, or even Twitter, a brief bio about yourself that will consist of your educational background and research interests, and the mentors can view your progress under the Activity tab, and the courses you are currently completing will appear under the Courses tab. Finally, after the completion of your course, the certificates will appear under the Certificates tab. So now you might be wondering what comes after completing the courses and example projects. The answer is possibilities are endless. For instance, you could go on to perform a research project like Ghazal Bhargava, a bioinformatics graduate from Amity University, Rajasthan, who completed a research project in the field of oncology. She uploaded a project on the Learn portal where an international community of students, peers, and bioinformatics experts can watch her presentation recording, read through her research project, like, share, and comment their feedback under the comment section. In the chat box, I am pasting the link to her project so that the participants can take a look. And another interesting student project outcome can be seen in case of Dilara Daikin, a molecular biology and genetics graduate from the University of Turkey, who completed a project in the field of astrobiology, which she went on to present at the Biomics Club session, a session where an international learning community of bioinformatics enthusiasts come together to share their work, debate on important topics, and share their queries to stay up to date with latest advancements in the field. You can take a look at her session recording in this link. I'm pasting it in the chat box. And in addition, she also went on to publish her project as a preprint on bioRxiv that she can share with her peers, mentors, and potential employers for future connections. And the link to the publication has been pasted in the chat box. Further, you also have the opportunity to participate in poster presentation competitions as a part of the Omics Research Symposium, like Dr. Umar Yadav, who worked on a machine learning project for which she won Best Technical Research Project as well as exciting cash prizes. And this symposium is an international research symposium that brings together experts such as Dr. Gareth Trebill and Dr. Joseph Borg, and among so many other experts who join as guest speakers and judges. And here's a link to her testimonial video where she talks about her project as well as winning the prize. The students are not just limited to graduates and doctorates. For instance, Andre Mordeno Sosa, a high school student at Mexico, presented his neuroscience project at the Regeneron International Science and Engineering Fair for which he won Potamkin Prize and the 4th award in the biomedical and health sciences category. And here's a link to the blog where you can read more about his work. All right, so now to view the various courses and example projects and student project, let us begin by signing up on the Omics Logic Learn portal. So for those of you who are signing up for the first time, here is the link for signing up. Please click on the link and I'll walk you through the process behind signing up on the portal. So once you click on the link, this is what you'll be able to see. So to create an account, you can simply click on this and then it will ask you to enter your name, email ID, and your password. And you also have the option to create an account using any of your existing social media accounts. That includes Google, Facebook, Apple account, GitHub account, or even Twitter. And with that, I'd like to pause for a couple of minutes so that the participants who have not signed up can sign up on the portal. And once you have completed your account on the portal, please leave a yes in the chat box so that I know I can proceed further. And if you already have an account on the portal, please leave it done in the chat box so that I can get a rough estimate of who all have already signed up. And with that, I will pause for a couple of minutes. Okay, so we have a lot of people who have already signed up. And if you're facing any technical issues, you can leave them in the chat box as well. All right, thank you so much. We have most of the participants have signed up. So since I also have an account on the option, I'm just going to click on the login now option here and sign in using my existing social media account. And once you sign in, you'll be able to view the various courses and example projects. So suppose, let's say you have a specific field of interest. For that, you can just enter the keyword that you're interested in. So let's say you're interested in machine learning. So please, you can just simply enter the keyword machine learning. And you'll be able to see the various courses and example projects that makes use of this concept. And now coming to the part where I was talking about updating your profile. For that, click on the tab that says welcome back with your email ID. And it is important to update your profile since the community manager and the mentors of the program will be tracking your learning progress. So how do we update your profile? For that, you have to just click on this button that says profile. And here, update your profile image, a brief description about yourself that will consist of your educational background and research interest. And it is also important to keep your name with appropriate capitalization. And you also have to link your social media handles as well. In my case, I've updated my LinkedIn account and my work ID. Now, this is the part where the community manager and the mentors of the program will be able to track your learning progress under the activity tab. And as explained previously, the various courses that you start taking will appear under the courses tab. And once you upload a project like Ghazal Bhargava, it will be displayed under the projects tab. And once you enroll for any of the programs, such as the research fellowship program, this will be displayed under the programs tab. And once you complete any of the courses with appropriate assignment and quizzes, the certificates will appear under the certificates tab. Now, you can also browse through and navigate the various student projects that you're interested to look at. This consists of a wide range of topics from oncology, infectious diseases, agriculture, astrobiology, and so on. So you can navigate through at your own pace and take a look at it. So here also you can just enter the keyword that you're interested to browse through. So let's say oncology, and that will display the various courses that makes use of the topic. So without further ado, now I'd like to pass on the stage to Nivedita Aranto. She is our research fellow, and she has completed a master's in biotechnology from Amity University and has joined the research fellowship program once again to publish her research project in peer-reviewed journals. So without further ado, over to you, Nivedita.
Thank you, Shrigavi.
Yes, Nivedita. Could you please share? No, that's fine.
Yeah, I'll just share my screen.
Thank you so much.
So I believe my screen is visible.
Yes, it's
Uh,
perfect. Thank you.
Hi everyone, my name is Nivedita Arambam. I completed my BSc and MSc in biotechnology from Amit University, Noida, and I joined Pine Biotech as a research fellow in December last year, and I've completed a project, and I rejoined to complete my process for publication. So my dissertation was, I did it from Manipur University. It was based on wet lab. It was finding out how a particular gene in HeLa cell line would uh, add to the progression of cancer. So it was wet lab, but then I had interest in dry lab as well, and I wanted to learn more about it, that too in oncology. So I joined Pine Biotech as a research fellow. Dr. Raghavendran was my guide, and he's still guiding me for the process of publication. So initially, we were made to complete some courses, which Srigori has shown us. Those were Python molecules, in Python, BioML, and Introduction to Bioinformatics. These are all basics courses that needs to be completed in order for us to move ahead. So you can do bioinfo in R or bioml in R. That's upon you, whichever programming language you're interested in. And after learning this, as I wanted to do in oncology, I had to decide which type of omics I want to go for, either genomics or transcriptomics, proteomics, or any other. And I chose transcriptomics because I wanted to work on RNA transcripts and gene expression. So I completed a transcriptomics course, which gave us a proper and clear idea about how RNA transcripts can be used to analyze and processed in a way that we would be able to find out how genes are expressed differentially in different types of conditions, in different conditions. And based on that, I started my independent project. So my independent project is based on relapse in AML, which is acute myeloid leukemia, in two treatment conditions. As you can see in the image, we have the allogenic HCT, which is hematopoietic cell transplantation, and another one we have the intensive chemotherapy. So these two treatment conditions are being used rigorously in AML as of now, and they are quite effective as well. But the problem is relapse of AML is inevitable. So I wanted to study how these two treatment conditions and their relapses has differences in the level of gene expression. That is the first purpose of the study that I wanted to find out. And I also wanted to find out how in transplantation the AML would affects the neurological processes, because I found that many neurological disorders were implicated. So I wanted to study further. And for that, the transcriptomics courses helped me. And right now I'm working on making the paper fit for publication. So I've completed the project, and the analysis and everything, but I am preparing the paper because I need to change writing and the images so that it is fit for publication. I'm also searching for publication platforms such as uh, journals through the platforms such as Elsevier, Web of Science, and Nature, to see what kind of journal and which impact factor would fit my quality of an area of the paper. I'm also working on making the supplementary section and the images and tables that need to be inside the paper. And I wanted to go for open closed access publication, because for open access I'd have to pay some fee, and that would be huge. So I wanted to go for closed access. And my end goal is to apply for higher studies because I want to go for a PhD in the area of oncology. I wanted to implement both wet lab that I did in my dissertation and dry lab that I learned from here to build up a research proposal so that I can apply for my higher studies. That's my journey. Thank you.
Thank you so much, Nivedita. And as research fellows who might be interested to join, like I have few queries for you. That is, even when I was starting out with the research fellowship program, I had this doubt of how can I narrow down and focus on specific data set, type of data set, such as should I choose the transcriptomics, should I choose genomics? So you were able to narrow it down to transcriptomics. So could you please share how you were able to do that and how one can go through a similar process?
Um, I
It looks
Hello.
like you're, yes, right.
Yeah.
Yes.
Sorry for that. I chose transcriptomics because I wanted to study the mRNA transcripts uh, of how the gene expression would affect. Like I wanted to study more into the biological pathways. So I felt that studying the gene expressions would help me understand how the biological pathways are affected by cancer. So uh, that's why I choose transcriptomics.
Right. And also, like, you have chosen the field of specialization as oncology, right? And in oncology
Yeah.
itself, there are lots of cancers that you could work on, like breast cancer, lung cancer, and so much. And your field of interest is AML. So, how did you come into that conclusion that you want to work with in the topic of AML?
Um, I believe at that time there were a lot of research papers that were written and talked about in AML and how the relapse are still an issue. So I thought I would work on AML and try to understand the relapse in these.
Right. And during the part of your coursework and example projects, were there any example projects that you really liked? And if so, like, why did you like it?
Example project. So I went through PDX, I believe, uh, because it was related to cancer. So I through that project.
Right. And then how did you find the mentor for this program? And how do you come to know about this program in general?
Um, I I came to know about it through LinkedIn, and I was assigned Dr. Raghavendran, and he is prominent in cancer studies in bioinformatics, I believe, uh, and others as well. But I was assigned Dr. Raghavendran, and he is a great mentor. Yeah.
Right. And there is also often a stigma that when you're completing a research paper, it's mostly if you want to transition into an academic field. Do you think that completing a research paper will be helpful in if you want to go in an industry related field, industrial research or so on?
Yeah, I believe that would help as well because in industry you people do data analysis as well. So that this will help them or guide them in a way on where to start off. So
And how do you plan on using your existing research? You plan on getting the research project published, right?
yeah.
And so after publishing, how do you think it will help your career? Like, which track are you planning to opt? Do you want to work in academia or you want to go into industrial research? How is it?
I want to go into academia and I want to pursue my PhD. And I believe these would add to my CV for application and the area that I want. So I believe while writing for my research proposal, and also uh, the relation to my area of interest is in cancer. So I believe this would help me out.
And the second last query that I have for you is, along the way that you are completing the program, were there any mistakes that you made and that stuck with you throughout the program experience that you would like to share it with other research fellows when they are enrolling for the program?
Stuck as in,
For example, in my case, when I enrolled for the program, I feel like I didn't go through the coursework as much seriously. But then when I started taking up the example projects, I felt that the theoretical background was very important because a lot of the theoretical concepts that are covered in the coursework is what that will be used in the example projects in an applied way. So that was one of the learning mistake that I had. So similarly, do you have any such tips or advice for the students who are just enrolling?
yeah, I do. So I opted for BioML in Python and bioinfo in Python as well, right? So I kind of skimmed through the examples and the answers, but like I felt like I should have tried to run it on my own more thoroughly and try to understand it more so that I could work with Python more efficiently. That was one of my problems that I faced.
Thank you so much. And finally, would you like to recommend this program to anyone?
Yeah, I would. I'm still on it, and I would recommend to anyone who would want to start out with omics data analysis, whichever career path they want to take, but who wanted to include omics into their studies. I would recommend for those people.
Thank you so much, Nivedita. Now, before moving on to the next part of the webinar, we'll have a short poll, and I would like to request Sparsh to pull up the poll so that it'll be an interactive session to make it more of an interactive. Yes, so the questions will be displayed in front of your screen. So just answer your interest-wise, like the questions that are being displayed on in front of your screen. So I'll just read them out very quick. That is, how important are bioinformatic analysis tools, skills, and resources in your current field of work? Is it extremely important? Is it very important? Somewhat important? Not so important? Not at all important? And the second is, do you find that an online learning community is helpful in keeping you motivated during your learning experience? And that would be significantly helpful, very helpful, somewhat helpful, or not at all helpful. And the third question is, are you interested in completing a research project and getting it published in a peer-reviewed journal? Yes or no? And what topics are you interested to work on for your research project? That is genomics, transcriptomics, metagenomics, epigenomics. And final question, how did you know about today's webinar? Was it through advertisement, organic social, email marketing, or through referrals? So I can see that 23 people, 10 people, 11 people out of 34 have participated. It would be great if you could fill it out as well, rest of the people, so that based on which Sonalika can also start recommending the courses for you that will be useful to you in enrolling for the program.
Are we good to end the poll, Shrigori? Or shall we
Yes.
wait for a few more seconds?
Yes, we'll wait for a few more seconds. Participants, it would be great if you could fill it out quickly. All right, then over to you, Sonalika.
Thank you so much, Shrigori. Just a second. Yes. So today I would like to share some of the resources that a research fellow gets the access to, how you can leverage these resources throughout your training and research period in the research fellowship program. And also we'll discuss about some of the research fellowship tracks that, or I'll call them specialization tracks that you can work on as a part of the program. So, as Sri Gauri has already mentioned about the Omics Logic Learn portal, I'll quickly go through some of the resources on the portal. So many a times people get confused about Omics Logic and the server. So the Omics Logic is the training portal where you have various resources put together, various courses on multi-omics and data science, and you can go through these resources to build an understanding of concepts and also learn how to run various pipelines or do the hands-on practice on the T-BioInfo server. And T-BioInfo Server is the hands-on practical server where you are going to run various pipelines on different omics data analysis. So I'll just quickly showcase you how you can navigate via the courses and then apply the knowledge that you gained from the Omics Logic Learn portal to run these pipelines on the T-BioInfo server. So when you click on under the courses tab, you have a list of courses that you can go through. Now, how do you figure out which specific course you have to go through and why am I talking about a coursework here? So as Nivedita also mentioned, when a research fellow joins the program, it's very important for you to actually build the concepts and know what you have to work on and what should be the pipelines that you should be running and what is the basis behind them before you actually start working on your research project, before you actually start applying those concepts to those pipelines. So as a research fellow, when you join the program, you go through an intensive training process in the first month or in the 2 months if you join the 6 months program. So the first month is for your coursework where you have to go through a specific specialized training. And for that training, you need to pick some courses from the portal itself. So in the search bar, if you simply type, if you select the courses tag from here on the right side, all of the courses will be listed out. To get started, those who come from a non-bioinformatics background, you can go through the first coursework, that is Introduction to Bioinformatics. This specific coursework will help you understand what the field bioinformatics is all about. It's a very basic short coursework and you will be able to complete this in just a day or few hours maybe. So going through this coursework will help you understand what is the role of bioinformatics in healthcare, what is big data in bioinformatics, what is translational bioinformatics, what are the various opportunities that you can leverage in this field. And the next coursework that you can go through just to figure out which specific field would you like to work with is bytes and molecules. We understand that as a student, if you are a bachelor student or a master's student, you might not have a specific research interest, or I'll call a specific research topic that you would like to work on. And you also might not know what are the various omics data types. In your academics, you are mostly introduced to fields like genomics, proteomics, or structural biology. But what about transcriptomics or metagenomics or maybe epigenomics? So this is the coursework bytes and molecules that we have put together for you to get introduced to different omics data types and also about some research specialization tracks where you can apply these different types of omics data analysis. Now, once a research fellow completes these introductory courses, you will be then able to pick a specialized coursework maybe after having a research interest. For example, if you are a research fellow and you complete these two courses, after completing these two, you might get an interest in one of these omics data types that you would like to learn about more. So you then take up the specialized coursework too. Now, I would like to this muted. All right. So now let's say from bytes and molecules coursework, a research fellow gets interested in the transcriptomic specialized training, we recommend them to take up the specialized training. So the training process is divided in 3 parts. The first part is an introductory phase where you go through these introductory courses to figure out which specific specialization track would you like to pick, or which specific type of data analysis would you like to learn. And in this coursework, how are these courses now arranged? That is what I would like to show you. Here you have some concepts explained about a specific type of data analysis, and you also have the guidelines to run pipelines on the T-BioInfo server. You have a mix of coursework put together here, some content, some videos and quizzes and assignments to also check the concepts that you gain throughout the coursework, are they correct or not. And now let's quickly jump on to the T-BioInfo server, about which I have been mentioning for a while now, that you have some guidelines put together here, which mentions how you can run the practical hands-on using transcriptomic data analysis on the T-BioInfo server. Why am I using transcriptomic word here is because, yes, I have opened the transcriptomics coursework. So we have the steps or the guidelines mentioned here in the coursework that you have to follow. Now, what is the T-BioInfo server all about? So let's quickly jump onto the T-BioInfo server. This is the hands-on practical server. Now, on the T-BioInfo server, you can see various sections put together. Today in the Research Fellowship webinar, I wanted to showcase you these different sections of the T-BioInfo server and also let you know how easy it is to analyze big data and visualize it through interactive visualization techniques or methodologies, and also let you know how you can learn it easily via Omics Logic Learn Portal resources. Thank you, Ujasvi, for sharing the link in the chat box. So on this server, you will find different areas of analysis put together: transcriptomics, genomics. Then you also have metagenomics section, machine learning and data sciences over here. You have phylogenetic analysis and single cell analysis. I would like to quickly give you a demo on one of the concepts that is called differential expression. Now, it's not important that most of the keywords that I'm using here, you might be familiar to it as a graduate or a master student as well. But throughout the training duration, you will be introduced to these keywords, so you need not worry about them. All right, so now let's look at one of these demo pipelines just to understand how easy it is to run these pipelines that I have been talking about and also visualize some of the results. Now, what are pipelines? Now, these pipelines are basically some sets of algorithms or tools that have been put together, and a pipeline follows a path. That is, you have to select, you start off and then you select certain number of tools and you set some parameters and then you end that specific pipeline that you began. That is what is called in very simple terms, a pipeline. I have made you understand this in a very layman language. Now, this is a demo pipeline. In a demo pipeline, you already have the data uploaded at the backend, and this demo pipeline has been put together for you just to see how easy it is to run the pipeline with these different tools. So this is a differential expression pipeline where you just want to look at some differentially expressed genes or genes that have an abnormal expression level, and then see what is the role of these genes in different pathways that lead to a specific disease or a disorder. So let's quickly run this demo pipeline. Now, when you talk about the hands-on part on the T-BioInfo server, I just mentioned that you have to upload a specific type of data, and in this demo pipeline, it's already uploaded. But when you run your own pipeline, it becomes very important for you to first of all understand what is this data that we are talking about. What is the format of this data? From where do you extract this data? And then how do you prepare these input data files to run a pipeline? After that, how do you actually run the pipeline and set the parameters? while opting or choosing these different tools that we have put together here. So all of these steps are mentioned again where it's in the coursework that you have to go through and understand first of all. So I'll select some tools here. I selected STAR, then DSEC for differential expression. And for pathway annotation, I'll select enrichment analysis and GSE. And I'll simply end this pipeline. Now, when I end this pipeline, I can see some visualizations that we'll have a look at. And also I have some results to download and also view. Now, these include some TXT files and also some plots and graphs that would help us interpret our results. So here is the process that you just saw that I also mentioned, from taking the data from various public repositories to actually interpreting some biological insights out of this data. And throughout this research fellowship program, it's important, first of all, for you to understand all of these concepts, the flow of the pipeline, the algorithms involved, the parameters to be set, and then see how to interpret these different results. Once your training process is complete, then you will be proposing a research question that you would like to work on and start off with your research question. Let the files download and let them open. Now let's quickly see what do these visualizations look like. So this is a heat map. I'll just close this. This is a heat map where you can see which all specific samples were involved in these different, or we can say it like this, that which all are the different types of genes which have abnormal expression in these different samples. All right, and I won't be going into much depth of these visualizations right now, but I just wanted to show you how interactive and easy it is to look at these plots. Now, this is a volcano plot, which shows you which are the genes with abnormal expression level. You have these genes listed here. You can see them when you hover over the points. And then you have a dashboard where you have the heat map here with the gene expression value. Also, you have these genes and genes in respective samples and their expression. And if the expression is abnormal, and then you have the volcano plot. And then you can also pick a specific gene and look at the level of expression of a specific gene in different samples. So this is how you can interact with your results and interpret the biological insights. And if you talk about pathways, then if you click on the enrichment tab here, then you can see which all genes that you just saw are differentially expressed. They're involved in which pathways. And again, you have the gene ratio here and the expression as well. So that is how easy it is to actually work with data. Now, when you join the research fellowship program, we understood that you have to go through a specific training or let's say a specific coursework in a specific type of omics data analysis. It could be transcriptomics, it could be genomics, it could be metagenomics or epigenomics or structural biology. Now, when you are well versed with a specific type of data analysis and you have to then pick a research question, the question arises here, which specialization tracks can you work on? Which are the different research areas or research fields that you can work on? So to help you with that, we have put together a page that lists various research project topics that you can pick from. We can share this page with you. I'll briefly go through the page in the webinar today. Won't be going into much depth right now as we are short of time. So the link is in the chat box. You can go through it, go through each one of these questions that you can pick to work on in the research fellowship program. Now, we basically work in 6 different specialization tracks. The first one is infectious diseases. Now, on this page, what we have done is we have listed the specialization track, and then we also have the different types of omics data analysis that is possible for each of these specialization tracks. And under each one of them, we have the research questions present that you can answer or that you can pick for your research question. All right. So the first is the infectious diseases track that you can work on. And then we have the different types of data analysis possible present over here. You can take your time and read through each one of these questions that you would like to answer. And then we have the second specialization track, that is oncology, and again, different types of data analysis types possible. Precision medicine is the other type of specialization track that we are working on. Then we have agriculture, and then we have neurosciences, metabolic disorders, and space omics. So you can go through the research project topics over here that you would like to answer as a research fellow in the program that could assist you. And I think that was it that I wanted to share. Two different resources that you can leverage throughout the research fellowship program, and also the specialization tracks that you can work on after your training is completed. So that was all. If you have any question, please put it in the chat box so that I can answer it for you.
Thank you so much, Sonalika. So does the participants have any queries? Please put them in the chat box if you're having any queries regarding the specialization tracks or any of the resources that was explained now. If not, I'd like to invite Sparshkar to talk about the registration cost and how one can enroll for the research fellowship program. So over to you, Sparsh.
Thank you so much, Sonalika, as well as Sri Gauri. So let me share my screen and I'll quickly explain how one can register and what are the resources one would have access to when they enroll in this particular program. Okay. So could you please confirm, Sri Gauri, if my screen is visible?
Yes, we can see the research fellowship program page.
Perfect. So this is the research fellowship landing page, and I'll quickly share the link here in the chat box. Thank you so much, Ojasvi, for sharing the link. So any participant who is interested can scroll with me on this particular page. So this research fellowship program is designed for everyone. So there is no constraint that a particular student of a particular level of education can only enroll in this particular program. We have participants across globe who are either in their undergrad, grad, or maybe in their post-graduation, or also there are faculties and researchers who have been enrolled in this particular program, and they are like researching on different domains, on different area of analysis. So this program is for all those young researchers and students who want to take advantage of bioinformatics, computational biology resources, and want to analyze complex life sciences data and become well versed in bioinformatics and multi-omic field. Right. So in this particular program, we will be providing you with some good resources. So the first resource is Omics Logic Learn portal, which Sri Gauri have already discussed in the beginning of the presentation or the session, I would rather say. So in this particular Omics Logic Learn portal, the name is self-explanatory. So there are n number of courseworks, lessons, example research projects, or case studies on different domains, on different area of analysis, which one can go through and leverage. Then you will have training, then you will have mentorship, then you will have access to the T-BioInfo Server, which Sonalika just showed, and then different area of analysis, which we have on our platform, through which you can learn, through which you can analyze, and through which you can interpret further, both statistically and biologically. So in this research fellowship program, we in a very layman language, I would say, we have divided this research fellowship program in 3 stages. So the first stage is to get you trained. The second stage is the analysis part, the literature review part, or the data set or methodology finalization part. And the third stage is to jot it down whatever you have done in your analysis part, right? So the first part, that is the training part. Any like Shrigori, could you please see if someone's mic is on? Uh, okay. So yeah, so I was saying the first stage is the training part where one would go through with different courses, different example research projects or case study according to their research area, according to their research interest and their research question. So this particular stage, we'll be doing this via the help of Omics Logic Learn portal. Once your training is done, you will jump onto the second stage, that is your analysis stage. And analysis stage includes literature review. After reviewing later literature, you will be finalizing a particular research question on which you'll be working, then a particular data set on which you'll be working and analyzing further, and prepare a particular methodology. And following that methodology, you will come up with the results and do the biological and the statistical interpretation further in the end. So this whole process is either done on the T-BioInfo server through running pipelines on different area of analysis, or through the like coding part like R or Python, or where we can include machine learning or data science aspect into it. And then once it is done, you will be shifted over to the third stage, that is the report writing stage. So whatever you have done in the second stage, your analysis stage, you'll be jotting down in a proper scientific manner, a proper scientific manuscript, and then further publish it. Okay, so as you can see, so these are the topics which we'll be covering in the training part, that is introduction to bioinformatics and the basics of data in life sciences. So if someone is beginner in bioinformatics, so there is a particular course for that is introduction to bioinformatics, through which we'll be helping them and guiding them to know what exactly is bioinformatics, the terminologies in bioinformatics, different applications of bioinformatics in healthcare, in translational research, etc., and how bioinformatics and computational biology tools can help in cutting edge research. Now, the omics data types like genomics, transcriptomics, metagenomics, or epigenomics, then there are data science and machine learning aspects for non-engineers and biologists, then R and Python programming is there. Now, when we move on to second stage, that is the literature review and plan out your project, as I discussed briefly. So there are few steps, there are few things which you need to remember. One is make your research question compelling to those already in the field. Second, set specific parameters to answer the research question conclusively. Third, find data sets that fit your objectives and explore them for batch effects or bias. And fourth, get feedback from our expert mentors on your topic and analysis proposal. So these are few things which you need to keep in mind when you perform literature review and plan out your whole analysis part, your whole methodology. And next, what are the potential outcome? So now you have completed your project, now your research work is done. So what are the potential outcomes of this research fellowship program? So number one is expert feedback and overall evaluation of project quality, innovation, and analysis execution. The second is poster presentation at the research fellowship meeting or a research symposium. For a third is path to collaboration with expert mentors or faculty at partner university. And fourth is path to a full publication. So these are some potential outcomes of this research fellowship program, which one can have, leverage through. Now, there are three different tenures, rather I would say four different tenures in this research fellowship program. One is of 45 days, another is 60 days, then is 3 months, and then is 6 months. So what is the difference between these all tenures? There is no difference, to be very precise, there is no difference. It's just the analysis part. So in 45 days and 60 days, there will be a kind of an intermediate level of analysis. You'll be maybe like doing only the literature review part and preparing a project kind of report. So whatever you have done in the literature review thing, that's all. And very minor reviewing would be there, very minor analysis would be there. When it comes to a full-fledged research project, so one should either choose the 3 months or the 6 months. So in the 3 months or the 6 months, we'll be following the pattern that I just explained, the stage 1, stage 2, and stage 3. So the resources which one would have access to are the asynchronous courses, the hands-on assignments, group sessions, program course access, omics logic program access, then educational cloud pipelines, the T-BioInfo Server platform, online mentor support, online technical support, access to live group sessions, etc. So these are the resources of all the 4 tenures. And the outcomes are, for 45 days, it's a literature review and a project report. Then for 60 days, it's literature review, project report, and a research poster. For 90 days, and then for 6 months, it's literature review, project report, and a research publication. Okay, now if someone is very much interested, so in this particular page only, we have provided with the payment links for all 4 tenures. And right now we are providing everyone with a 20% scholarship, and the coupon code for that 20% scholarship is take 20. And I have pasted the coupon code in the chat box. If anyone is interested, they can simply visit this page and click on which particular tenure they want to enroll for. And if they want to complete a research full-fledged project and come up with a research publication, or if it's a like kind of a literature review and prepare a project report only, according to your wish, you can click on this payment link. It will be redirected to the purchase part. You can fill out your credentials of your card and then write the coupon code and then pay now. So so this is how simple you can do the payment part. If further I'll scroll down, you can see there are different students projects on different domains like infectious diseases, space omics, precision oncology, machine learning, skin disease, data driven drug discovery, etc. And as I go down, you can see our different collaborators from all across the globe, and the students from all these collaborators have been a part of research fellowship program, or like they are right now also doing their research fellowship program with us. So this was a brief description and also the mentors of this program. So we have industry and academic mentors, and they'll be guiding you according to the expertise of their. So we have a separate mentor for cheminformatics and drug discovery, then a separate mentor for metagenomics or genomics part, then a separate for our Python or data science or machine learning thing. So this is how everything goes. And these are some blog posts about the student research projects. So this was all about the registration, the resources, and the payment, how one can do, and also the scholarship. So if anyone is having any query regarding the registration part or the scholarship part or how one can avail, please feel free to ping in the chat box and I'll be there to guide you how one can proceed with the payment checkout process or any query, any kind of query. So thank you. Over to you, Sri Gauri.
Thank you so much, Sparsh. So in the chat box, I'm pasting Sparsh's contact number and her email ID that you can reach out. The contact number is also available on WhatsApp, so you could text her as well. So with that, does the participants have any queries about registration or the specialization tracks that were discussed? If so, you could either try to unmute yourself and talk, or you could also share your queries in the chat box. I'll pause for a few minutes. And if not, we'll be ending the meeting soon.
I guess Sri Gauri, there are no more questions. So
All right. Yes.
Yeah, if someone is having any kind of question regarding the scholarship or the registration part, please feel free to ping. Okay, so Rekha is asking something. Ma'am, what is the registration fee? So Rekha, could you please confirm if you are from Government City College, Hyderabad, if I'm not wrong? Perfect, Rekha. So, Rekha, could you please DM me your contact details so that one of our intern would contact you and guide you about the registration fee, because we have a specific discount, or we can say a specific scholarship for the partnership, like the partner or the collaborators. And Government City College is one of our collaborators. Yeah, thank you for the email address, Rekha. Yeah, that's more better. So I have noted down your email address, and one of our intern would definitely call or ping you. Yeah, they will guide you shortly.
Thank you so much, Rekha. Does any other participants have any further queries related to scholarship or discount? If so, please put them in the chat and we'll be happy to help. Otherwise, thank you so much, everyone, for today's participation, and have a great day. Thank you.